Hi, this is the recording to accompany the slides for the third of 3 lectures on the digestive system. Let's begin. So in this lecture, what we're going to go through is we're going to go through these 3 accessory organs and then talk about digestion and absorption as an overview. So we're going to start off with the first accessory organ, which is the liver. Liver is the largest gland in the body, located just underneath the diaphragm over here, protected by the ribs. The functional unit of the liver is the lobule. So it's hexagonal in shape, so 6-sided. And what you can see here is you've got a central interlobular vein with radiating pathways from it. In terms of the liver, it also has an exocrine function in that it produces bile, which is released into the duodenum, the first part of the small intestine. When we look at the function of the liver, you can see that there's the liver is involved in quite a lot. So it detoxifies toxic substances that come through either through the systemic circulation or through food. It produces bile, which is then released into the small intestine, and bile contains cholesterol. It breaks down and recycles old red blood cells, right? And that's also the function of the spleen, if you can remember. And when it breaks down old red blood cells, then iron and bilirubin are extracted from it. The liver receives blood directly from the digestive tract, so it receives a lot of nutrients that are absorbed from the small intestine, and so it metabolizes and stores those absorbed nutrients, and it forms, also synthesizes fats, proteins, and stores carbohydrates. One of the things that the liver produces is plasma proteins. So in terms of the important plasma proteins that it produces fibrinogen and plasminogen that's involved in the clotting process. It produces angiotensinogen that's involved in the renin-angiotensin-aldosterone system. It produces albumin that's involved in maintaining blood colloid osmotic pressure. So it produces a lot of different proteins. And again, if you look at the first 3, you have the ogen at the end, which means that it produces it as inactive components so that it can be activated when the need arises. As well as storing nutrients, it also stores iron and vitamin D and glycogen. And remember, glycogen is a polymer of glucose subunits, right? So it's a string of glucose subunits, and it stores it as glycogen because it can store it more compactly. And when we need an increase in our blood sugar levels, then what happens is glycogenolysis occurs to break down glycogen and release the glucose monomers into the systemic circulation. In terms of the organization of the functional units of the liver, they're called lobules because they're in the liver. They're called hepatic lobules. So hepatic refers to the liver. So the hepatic lobules contain hepatocytes. So cyte is cell, hepato refers to the liver. Hepatocytes are the liver cells that are involved in the main functions of the liver. So it produces bile, it synthesizes and stores protein, fats, carbs, detoxifies drugs, and other harmful substances. So all of this are functions of the hepatocytes. As I said, the hepatocytes are organized in a hexagonal lobule around a central vein. So the central vein is over here, right? It's just in the center of each of these lobules over here. And around them, we've got capillaries, which are sinusoidal capillaries. And if you remember what sinusoid capillaries are like, they've got a incomplete basement membrane with gaps in between the endothelium. And these large gaps are large enough to allow proteins that are produced by the liver to enter into the bloodstream, and allows old red blood cells to exit the bloodstream in order to be broken down by the hepatocytes. As I said, the hepatocytes produce bile. Bile is not released into the blood. Bile is released into its own system, and the system is called canaliculi. We've seen that word canaliculi before when it refers to the connection between cells in the mature bone. So canaliculi connects osteocytes in bone. So the canaliculi here are little canals that allow the bile that's produced by the hepatocytes to be transported from the hepatocytes in towards the small intestine, right? So it delivers bile produced by the hepatocytes. The next thing are these portal triads. So the portal triads are found at each end of the hexagon. Right? So this is a portal triad, that's a portal triad, that's one, that's one, that's one. Right? So at each end of the six-sided hexagon, we've got the portal triad. So portal triad refers to three. So there's three things that make up the portal triad. So the first one is the bile duct that collect all the bile that's being delivered from the canaliculi into it. The second one are the portal venules and the portal arterioles. So again, we're going to look at that in the next slide, right? So, and then another thing that's found in the hepatic lobules are reticuloendothelial cells that are phagocytes in the hepatic sinusoids that help to remove bacteria, dead white blood cells, and foreign materials, right? So they work the same way as phagocytes anywhere in the body. And then the last thing is a central vein over here that collects the blood that go through the capillaries, the sinusoids, to return that blood to the heart. So you can see here each central vein then collects the blood into the interlobular vein, and the interlobular vein is connected between each lobules to direct the blood to the hepatic vein, and from the hepatic vein it goes back into the heart through the right atrium. When we look at the portal triad, as I said, there's three things in the portal triad. One is the bile duct that helps to collect bile from the canaliculi over here, right? The second one is the portal vein. That is a branch of the hepatic portal vein. So hepatic again refers to liver. So this one actually comes directly from the stomach and the intestine, and so contains absorbed nutrients. Now, because it's gone through the capillary beds in the stomach and the small intestine, the blood in the hepatic portal vein and then the interlobular portal vein is oxygen poor, right? And because it's oxygen poor, we need another source of oxygen in the blood. And so that other source of oxygen in the blood is found in the interlobular artery. That is a branch of the hepatic artery. And again, there's more information in the next slide, right? So the interlobular artery transport oxygen-rich blood directly from the heart. The interlobular portal vein transports nutrient-rich blood that is oxygen poor from the stomach and intestine, and the interlobular bile duct transports bile that's been produced by the hepatocytes released into the canaliculi. So in each hepatic lobule, you've got the blood moving from the portal vein and the artery through the sinusoids, where it goes past so over here, it goes over here, where it goes past the hepatocytes that do its job, and then it directs the blood in towards the central vein, when it is then collected and directed into the interlobular vein, to the hepatic vein, towards the heart. Whereas bile is produced by the hepatocytes. Whoops. But bile is produced by the hepatocytes, released into the canaliculi, where it's then collected into the interlobular bile ducts. So again, what we've got here is we've got two halves. One is the flow of blood in through the liver, and you can see here when it's 27% of blood of cardiac output goes to the liver. And what you can see here is that you've got two sources of blood. As I said, we've got oxygen-rich blood direct from the heart through the hepatic artery. And you can see here that this oxygen-rich blood reaches the liver at a very high pressure, but there's not that much blood, right? 0.3 liters at 100 millimeter mercury. Whereas the hepatic portal vein, you can see here, it's a lot of blood coming from the digestive tract, 1.05 liters a minute. And you can see here, it's low pressure. So low pressure because it's gone through the capillary bed in the digestive tract. And there's a lot of blood because we absorbed a lot of nutrients through the digestive tract, right? And so the two sources of blood combine. We've got oxygen to help fuel metabolism. We've got the nutrient-rich blood that is the source of raw materials to make fats, proteins, and carbs. They get processed by the hepatocytes, and then the blood then leads through the central vein of the hepatic lobule over here, in through the hepatic interlobular vein, to the hepatic vein, in through the inferior vena cava, through to the right atrium of the heart. So that's the flow of blood as it goes through the liver. In terms of the flow of bile, bile is made by the hepatocytes of the liver. It then is released into the bile canaliculi, where it flows into the bile duct in each of the portal triads. And then it's collected by the right and left hepatic duct over here. And then it exits the liver as the common hepatic duct. And then it merges with the cystic duct. So the cystic duct is the one that goes towards the gallbladder. And the cystic duct and the common hepatic duct merge to form the common bile duct. And from in the common bile duct, it then makes its way in through the small intestine. Right? So what we're going to do next is look at bile and then the gallbladder. Right? So bile is produced and released continuously by the hepatocytes of the liver, stored in the gallbladder, and then released into the duodenum, which is the first part of the small intestine. You can see over here, the liver produces 1 liter of bile each day. The pH of bile is basic, right, to help to neutralize the gastric acid. And the ejection of bile is controlled by the hormones cholecystokinin, which is secreted by the small intestine. Most of the bile is absorbed from the small intestine and then recycled. So what happens is it's released into the small intestine and then absorbed from the latter part of the small intestine, and it goes around and is recycled between the liver and the small intestine, liver and the small intestine, about 17 times before it is broken down and excreted in feces. In terms of the components of bile, it contains lecithin and bile salts, which help to emulsify, to break down fats into smaller particles. So you can see over here, you've got these larger fat globules that are broken down by bile in a process called emulsification into smaller fat droplets. And when it's broken down into smaller fat droplets, then the enzyme lipase can chemically break it down further, right? Bile also contains bilirubin, which is a waste product from the breakdown of red blood cells. It also contains sodium bicarbonate, which increases pH of contents in the small intestine for optimal function. And it also contains cholesterol and products of detoxification. As I said, bile is produced by the liver at a constant rate, about 1 liter a day, and it's released from the bile liver into the bile duct, the right and left hepatic duct, where it goes down the common hepatic duct, where it merges with the cystic duct, and then from there it goes down into the common bile duct. So what happens is bile is only useful when there is fatty contents in the small intestine, right? All other times, what happens is the bile that's produced by the liver gets stored in the gallbladder. And so what happens is as the bile makes its way down the left and right hepatic duct, it causes the sphincters of the papilla to contract, so that instead of bile going down over here, it basically moves up towards the gallbladder, where it gets stored in the gallbladder. The gallbladder doesn't have enough room to store a lot of bile, and so as bile enters into the gallbladder, what happens is water is absorbed from the gallbladder, and the bile that's stored in the gallbladder becomes more concentrated. So it becomes 5 to 10 times more concentrated than it is when it's produced by the liver. And again, when you look at it, the gallbladder can only store 30 to 50 mils of concentrated bile. And so again, it needs to be concentrated, right? It can't store a liter of bile that the liver produces each day. And then when we need bile, then what happens is the smooth muscle layers of the gallbladder contract, and then bile gets ejected back through the cystic duct into the common bile duct. Structure of gallbladder, similar to structure of other parts of the body. You've got an outer connective tissue layer, a middle smooth muscle layer, and then an inner epithelium layer, right? You can see the inner epithelium layer is made out of one layer of columnar epithelium to allow for easy absorption of water and ions from bile in order for it to be concentrated and stored in the gallbladder. The next one is the pancreas, and the pancreas has got both exocrine and endocrine gland, and I'm going to remove this picture before I post it. But what you can see here is you've got the gallbladder over here, and what you can see here is the gallbladder, when it releases bile from the gallbladder, it enters into the common bile duct over here, where it mixes with pancreatic juices. So juices that are produced by the pancreas here gets mixed into the ampulla over here, and the release of the pancreatic juices with bile into the small intestine is controlled by the sphincter of Oddi, which is also called the hepatopancreatic sphincter. If we eat a lot of food that doesn't contain fats, and so we don't need bile to be released, what happens is the pancreatic juices are released into the duodenum through this accessory pancreatic duct, right? We know that the pancreas has both exocrine and endocrine functions. In terms of the endocrine functions, we talked about it within the endocrine lecture. So it releases hormones such as insulin and glucagon to control blood glucose levels. But a lot of the a major portion of the pancreas is involved in pancreatic secretions, in exocrine secretions into the duodenum, right? And so the secretions are called pancreatic juices, also contains digestive enzymes. So when we look at the secretions of the pancreatic juices, again, pancreas produce 1 liter a day of pancreatic juice that's slightly alkaline, again, to try to neutralize the acidic pH. And once the gastric acid is neutralized, pepsin is also inactivated. Remember, pepsin is a hormone, is an enzyme released into the stomach that digests proteins, only active at very low pH. And so when the pH becomes higher, it becomes inactivated, right? And this pH is optimum for the final digestion that occurs in the small intestine, right? Control of pancreatic juices depends on the release of these hormones. So again, if there is a high fat content entering into the small intestine, then cholecystokinin is released to stimulate the secretion of pancreatic juices and the release of bile from the gallbladder. But if the contents of the small intestine do not contain a high amount of fats, then secretin is released, and that then releases juices that are high in bicarb and low in enzymes. In terms of what's found in pancreatic juices, we've got sodium bicarbonate, again, to increase the pH of the duodenum, the small intestine. We've got proteases that digest proteins. We've got, and again, the enzymes that digest proteins that are produced by the pancreas are released as their inactive forms. So you've got trypsin, ogen, chymotrypsin, nagen. You've got prolactase and some other enzymes that are not active when they are in the pancreas. We also have trypsin inhibitor that stops the conversion of the inactive trypsinogen to its active form. We've got lipases that digest fats, nucleases that digest nucleic acid, and we've got amylases that digest starch. So we've got a whole host of enzymes that help with the final digestion when it's released into the small intestine. So in this, we see why the pancreas doesn't digest itself. So the first one is that the enzymes that digest proteins are produced as zymogens or inactive enzymes. And what it does is when it's when it's produced as an inactive enzyme, it doesn't digest proteins. Trypsin inhibitor is also found in the pancreatic acinar cells, in the ones that produce exocrine glands. And what happens is that once it reaches the small intestine, Then we've got enteropeptidase, which is an enzyme in the small intestine that activates trypsinogen and converts trypsinogen into trypsin. Once trypsin is formed, then trypsin helps to convert all the other inactive proteases into their active form, right? So if you look at these enzymes, these are all inactive enzymes. You can see because they either have an ogen at the end or a pro at the start, and that means that they're inactive. And then in the presence of activated trypsin in the small intestine, it gets converted to the active versions, which then help to digest proteins, right? So these pancreatic proteases are activated by high pH as well as the presence of trypsin. So let's go and look at the overview of digestion and absorption. So in terms of digestion, there are two types of digestion: mechanical digestion that helps to break down food into smaller particles but doesn't change its chemical composition. And then there's chemical digestion that helps to break down food chemically from more complex chemicals into their simpler versions. And then you've got absorption, which is the movement of nutrients from the lumen of the digestive tract into either the lymphatic or the blood circulation. As you can see here, mechanical digestion occurs in all parts of the digestive tract, with the exception of the pharynx and the esophagus. Chemical digestion occurs where mechanical digestion also occurs, with the exception of the large intestine, where the only chemical digestion occurs because of the presence of bacteria, not of enzymes. And so what this means is that mechanical digestion can occur at the same time as chemical digestion, right? Absorption can also occur before mechanical and digestion is complete. So as I said, while 90% of absorption occurs in the small intestine, there's a small amount of absorption that occurs in the stomach as well as in the large intestine. So obviously, when absorption occurs in the stomach, chemical and mechanical digestion is not yet complete. And when you've got the digestive food or the undigested food and the unabsorbed food entering into the large intestine, then it leaves it as fecal matter, right? Anything that's in the digestive tract is considered to be outside the body because it's not yet part of the systemic or lymphatic circulation. In terms of the digestive enzymes, this is an overview. And so what you can see here is the blue are the ones that digest lipids. The purple is the one that digests carbs. The gray are the ones that digest proteins. And then the beige are the ones that digest nucleic acids, right? And what you can see is the asterisks are the enzymes that digest proteins that are released as the inactive version in order to stop autodigestion from occurring. In terms of absorption, there are different ways that things get absorbed. Over here on the top right, you've got the amount of water that's absorbed and water that's secreted into the digestive tract. So red lines, red arrows are where water is absorbed, and these sort of blue-green arrows are where water enters into the digestive system. So food and drink, 2 liters we're supposed to drink every day, saliva 1.5, digestive gastric secretions 1.5, and so on and so forth. And what you can see is the small intestine, when it absorbs nutrients, also absorbs water. So most water is absorbed in the small intestine. Some is absorbed through the colon in order to make our fecal matter not diarrhea-like. And then in feces, we lose a small amount of water when that happens. In terms of absorption, you've got different mechanisms of absorption. So active transport, passive diffusion, facilitated diffusion, co-transport, and endocytosis. Over here, we've got what the different types of nutrients and the form that can be absorbed through the digestive tract. So again, carbohydrates are broken down into monosaccharides such as glucose, galactose, and fructose, where it's easily absorbed. Proteins are broken down into simple amino acids, so single amino acids, 2 amino acids together, dipeptides, or 3 amino acids together, tripeptides. Fats are broken down into monoglycerides, glycerol, and free fatty acids, where it gets absorbed. And then nucleic acids are broken down into its components, where again it gets absorbed. When we look at carbohydrate digestion and absorption, we can see here the various enzymes that are involved in breaking down carbohydrates. So you've got there in a table form, as well as in a, mind map form. And what you can see here is this is the lumen of the inside of the tube of the digestive tract. And what you can see here is glucose moves in through the epithelial cell through a process of co-transport with sodium ions. So moves in the same direction using energy that's produced by the movement of sodium, and then it uses an active transport system again to actually move, so sodium moves into the interstitial fluid using an active transport system. Once glucose enters into the epithelial cell, then it can move in through the blood through facilitated diffusion, right? So it goes into the blood once it's absorbed in the small intestine. Next one is protein digestion and absorption. So again, in the stomach and in the small intestine, you've got a variety of enzymes that help to digest proteins. Over here, and then it's absorbed through, again, co-transport with sodium, same as the glucose before, and then it then is transported across into the blood capillaries, right? Once it gets transported into the blood capillaries, it goes to the liver, where it is processed, right? So again, it's absorbed to the liver, where it's processed over here. The thing with some of these protein digesting enzymes is that it's produced in the brush border, in the microvilli of the small intestine. So it's not just, so the protein enzymes aren't just produced by the stomach and the pancreas, but also by the microvilli of the small intestine. Next part is the lipid digestion and absorption. So again, almost all lipid digestion and absorption occur in the small intestine. Bile salts speed up mechanical digestion of lipids and are important to absorption of end products of lipid digestion. So you can see here, lipase is the enzymes that chemically break down lipids, and lipase is secreted by the lingual and salivary glands, by the stomach and the pancreas. All the same enzyme, just secreted by different parts of the digestive system. And then you've got bile salts that help to mechanically break down fat globules into smaller bits, right? And then in terms of absorption, it gets absorbed in the small intestine, but instead of entering into the blood capillaries, it enters into the lymphatic capillaries. So you can see here, they absorbed lipids enter into the lymphatic capillaries, into the lacteals. So let's go through this. So you've got this huge fat globules that are broken down mechanically by bile salts through a process of emulsification into smaller fat globules. When it's smaller, then the enzyme lipase that's released by the pancreas can actually chemically break down the fats into simpler components, into free fatty acids and mono and diglycerides, and it encloses those components into these micelles, right? So these micelles are empipathic, so they are hydrophilic on the outside and lipophilic on the inside. And so it helps to transport the free fatty acids in this way. The micelles then go in close to the brush border, where the fats basically leave the micelles and diffuse directly through the plasma membrane to enter into the epithelial cell of the small intestine. Once they are in the epithelial cells of the small intestine, you can see over here, with the help of the Golgi apparatus, it forms triglycerides, right? And the triglycerides, together with phospholipids, form these lipoproteins. And the lipoproteins then form these chylomicrons, which is water soluble. And so it actually allows these water soluble fat enclosed things, lipoproteins, to enter into the lacteals, where it gets transported away from the small intestine. So just to put it together, the digestive tract functions mostly under the control of the autonomic nervous system. Exceptions are we choose to swallow and chew, as well as to defecate. So these are all processes that are under voluntary control. Movement of ingested materials through the digestive tract involves a series of peristalsis that moves the ingested contents in from the mouth towards the anus, as well as mixing that helps with breaking down the ingested contents into smaller and chemically simpler components. And we've got mechanoreceptors as well as chemoreceptors to help with those processes, right? So the chemoreceptors act as taste buds to differentiate between the different nutrients that we swallow. And depending on the nutrients that we swallow, we release different types of secretions, both exocrine and endocrine. And then that helps with the overview and overall digestive processes.